Hello students, how are you? Today is the third lecture in the series of lectures where we are discussing about that how bacteria deal with oxygen, right? What is the relationship between bacteria and oxygen? And according to that relationship, bacteria can be subdivided in the above groups that we can decide that bacteria can be obligate anaerobes or bacteria can be aerotolerant anaerobes, facultative anaerobes, microaerophiles and obligate aerobes, right? So bacteria are subclassified into these 5 types according to the relationship of bacteria with oxygen. Now what are the 2 important relationships of bacteria with oxygen? Number 1, can a group of bacteria use oxygen for energy metabolism or not? Can a group of bacteria use oxygen for the energy metabolism or not? Right? Number 2, that molecular oxygen, when it reacts with biological system, it can produce highly reactive oxygen species, right? They are very toxic molecules. How bacteria deal with those dangerous and toxic molecules? Can bacteria deal with those oxygen molecule related toxic products or not? Right? In previous 2 lectures, we have already discussed number 1, energy metabolism of bacteria and their relationship with oxygen. What we discussed last time in the previous lectures, that basically bacteria can have aerobic respiration or they can have anaerobic respiration or they can have anaerobic fermentation, aerobic fermentation pathways for energy metabolism. Now, as I mentioned previously, that how bacteria produce energy and what is the relationship of oxygen with that energy production mechanism. That those bacteria which use molecular oxygen as final electron acceptor, right? Those bacteria are said to be bacteria displaying aerobic respiratory process, right? What is aerobic respiratory process? In aerobic respiratory process, actually when energy, you can say big molecules like glucose, when they are broken down, right, and during that process when energy is released, who accepts the final electron? That is oxygen in case of aerobic respiration. As I mentioned previously, that in aerobic respiration, glucose passes through the process of glycolysis and then pyruvic acid is made, which enters into Krebs cycle and then electron transport chain, right? And eventually electrons are accepted by the oxygen. If this is the metabolic pathway, that glucose goes through glycolysis and then products of glycolysis enter into Krebs cycle and eventually electron released are carried to the electron transport chain and eventually oxygen is utilized as the final electron acceptor. We say this kind of energy production is aerobic respiration. Then there are some bacteria, they have anaerobic respiration. Now, what is the difference in aerobic respiration and anaerobic respiration? The biggest difference is that in aerobic process, final electron acceptor is oxygen, and in anaerobic processes like anaerobic respiration or anaerobic fermentation, oxygen is not used as final electron acceptor. Rather than oxygen, some other molecules are used or atoms are used. For example, when we talk about anaerobic respiration, again this process is same. when bacteria are doing anaerobic respiration, they are doing the glycolysis, they are passing the products of glycolysis through the Krebs cycle. Even there is electron transport chain also, but here is the difference, that when there is aerobic respiration, oxygen was used, oxygen, right, was used as final electron acceptor. In anaerobic respiration, right, glycolysis is there, Krebs cycle is there, electron transport chain is there, but at the end, final electron acceptor is not oxygen. It is some other molecules, like it may be nitrates, or it may be sulfates, or it may be elemental sulfur, or fumarate, right? So it means in anaerobic respiration, there is no use of oxygen. Now we come to this thing, anaerobic fermentation, right? In future we will be using the word only fermentation, but we should remember this is also anaerobic process. It's the process where oxygen is not used or involved in energy production as a final electron acceptor. Now, how oxygen is not used in anaerobic respiration by bacteria, oxygen is not used by anaerobic fermentation, but what is the difference? Common thing is that they both of them don't use oxygen. The difference is that anaerobic fermentation, glucose goes through glycolysis, and after that process goes to electron transport changes. For example, from glycolysis up to pyruvic acid or pyruvate, but there is no Krebs cycle and there is no electron transport chain, and of course no utilization of oxygen. So what really happens in fermentation? Under the process of glycolysis, glucose is broken down up to pyruvic acid, and then it might be converted into some other products, like for example pyruvic acid, and final acceptor of the final electron is pyruvic acid itself. It is not oxygen, and it is not oxygen, and how it is different from the anaerobic respiration? Remember, aerobic respiration, anaerobic respiration, and anaerobic fermentation, right? Let me compare it. All of them have glycolysis, but when we use the word respiration, it means there is Krebs cycle and electron transport chain. But in case of fermentation, there is no Krebs cycle and no electron transport chain. So what really happens? That this produces very less energy, and anaerobic respiration also produces less energy or ATP. The best amount, the maximum amount of energy is produced by the breakdown of glucose through the aerobic process, right? So it means when I say some bacteria are aerobic, it means they are able to utilize oxygen during energy production as final electron acceptor. It means those bacteria are able to generate a lot of energy for their survival and for their proliferation. But when I say there is anaerobic respiration or there is fermentation, it means that energy which is produced per molecule of glucose is far less as compared to the aerobic respiration, right? So this is what we discussed mainly. I slightly reviewed it. Secondly, we have to see, so what will happen, that if I say there is a bacteria which has only a fermentation pathway, metabolic pathway only for fermentation or anaerobic respiration, and that bacteria does not have metabolic machinery for aerobic pathway, then what I will say, that is anaerobic bacteria. But if there is a bacteria which is able to utilize oxygen as a final electron acceptor while producing energy, right, then we will say that bacteria is aerobic bacteria, right? Now, and we also discussed anaerobic processes are evolutionary more primitive. They are not advanced pathway. The most primitive metabolic pathway to produce energy from glucose is anaerobic fermentation. A little advanced pathway is anaerobic respiration. A most advanced pathway which produces maximum amount of ATP by the glucose molecule breakdown, that is aerobic respiratory pathway, right? Now we come to another thing. So when bacteria are having availability of oxygen, the first thing we have to determine, can bacteria use the oxygen for energy production or not? If bacteria can use the oxygen as a final electron acceptor, aerobic bacteria. And if they cannot use oxygen as a final electron acceptor, we say they are anaerobic bacteria. Clear? Then second relationship with oxygen. First relationship was between bacteria and oxygen, that can bacteria utilize oxygen or not for energy production. Secondly, once oxygen is there around biological systems, oxygen can undergo partial reduction and produce highly reactive, highly dangerous radicals or molecules, right? We have already discussed that also, that if bacteria or biological system are exposed to the oxygen, right? Oxygen, when it goes under partial reduction, right? What it does, it produces superoxide radicals, it can produce hydrogen peroxide, and it can produce hydroxyl radical. We have already discussed in previous lecture that how these reactive oxygen species can be generated when oxygen is available and it is partially reduced. And once superoxides or hydrogen peroxide or hydroxyls or all of them are present, they are highly reactive. That is why we call them reactive oxygen species, right? Why we call them reactive? Because they love to react with DNA or RNA, nucleic acids. They can react with proteins, they can even react with the fatty acids, right? And damage these macromolecules and damage the cell. So we say that if bacteria are exposed to the oxygen, within the bacteria, there will be production of lot of superoxides, hydrogen peroxide, hydroxyls. It means production of lot of reactive oxygen species. Or we can say that bacteria will have oxidative stress. It is not something pleasant for the biological system to have such aggressive molecules, right? So we say that when bacteria face oxygen, right, there is one thing good and one thing bad. The good thing is that if bacteria has aerobic metabolic pathway for the energy production, it will produce a lot of energy. But the negative thing is that when bacteria are exposed to the oxygen, reactive oxygen species are produced which can damage DNA, RNA, proteins, fatty acids, and damage the bacterial membranes. They can damage the bacterial nucleic acids, they can damage the bacterial proteins and enzymes, and they can kill the bacteria. So if bacteria have to survive in oxygen rich environment, then bacteria should have some defenses against these aggressors. Then bacteria must have some defenses against these aggressive molecules, highly reactive molecules. And yes, if bacteria don't have effective defenses against this oxidative stress, then bacteria will not be able to grow or multiply properly in the presence of oxygen. If bacteria have to survive and multiply in the presence of oxygen effectively, then they must have some mechanism by which they should be able to keep on continuously destroying these reactive oxygen species to protect their own macromolecules from oxidative stress. And yes, those bacteria which are aerobic, which can multiply in the presence of oxygen, they do have defenses, right? What are those defenses? There are special enzymes which actually neutralize these reactive oxygen species. The enzymes are superoxide dismutase, The second group of enzyme is, yes, catalase, and third one is hydrogen peroxidase. Hydrogen, okay, I will write it down. Hydrogen peroxidases. Now, if a bacteria contains dismutases or catalases or even peroxidases, then bacteria can survive or even multiply in the presence of oxygen, because due to the presence of oxygen, whatever reactive oxygen species will be produced before these molecules can damage the macromolecules of bacteria. Bacterial enzymes like dismutases, catalases or peroxidases will destroy or neutralize these reactive oxygen species. How they will destroy, I have discussed in previous lecture. So up to now, what did we discuss? We discussed that when we are going to classify bacteria, that are they aerobic or anaerobic or in between different types, right? When we classify bacteria in these 5 types, the base of the classification is, the foundation of the classification is, number 1, how bacteria deal with oxygen. Can they use the oxygen for energy production or not? Are they aerobic bacteria or anaerobic bacteria? Secondly, if bacteria are exposed to the oxygen and reactive oxygen species are produced, oxygen toxic molecules are produced, oxidative stress is produced, we need to see, do these bacteria have any defenses, full defenses or partial defenses again for neutralizing these aggressive molecules? What is that? That we have to see how bacteria deal with reactive oxygen species, or do they have capacity to deal with reactive oxygen species or not. If bacteria can deal with these and neutralize them, it means they must have dismutases, they might have catalases and even peroxidases. But if bacteria are destroyed in the presence of oxygen, simply oxygen is toxic to them. The presence of oxygen is a poison for bacteria. It means that whenever such bacteria are exposed to the oxygen and reactive oxygen species are produced, they are unable to defend themselves, right? And reactive oxygen molecules, they destroy the macromolecules of bacteria. Why? Because bacteria don't have defenses against oxidative stress. It means such bacteria do not have superoxide dismutases or catalases or peroxidases, right? Now, when I discuss all these 5 categories of these bacteria, right? 5 these classes of bacteria, right? In every class I will discuss about that. For example, if I say there are aero-tolerant anaerobes, I will discuss what is their energy metabolism and how they deal with reactive oxygen species. Are they able to defend against these or they cannot defend against, right? Now, before really I go for that, I would like to introduce you that how, for example, if from an abscess, right, or from pus, we take sample, specimen, and in the laboratory in a culture, we basically cultivate the bacteria in a culture medium, right? How we can, depending upon culture media, how the bacteria grow, the pattern of growth of bacteria in that culture medium help us to differentiate these 5 categories. What do I mean? I mean that now I will introduce you to a special type of culture media, in which if even these 5 of these bacteria are put together in that culture medium, all of them will grow in that culture medium, making a different growth pattern. And by the growth pattern, of bacteria within that culture medium, we should be able to decide that what kind of bacteria are these? Are they anaerobic or aerobic? Are they aerotolerant or microaerophilic? So now we are going to talk about those concepts. But before that, I will introduce what is that culture medium. Now, to have a culture medium in which such kind of bacteria can be grown, all of them can be grown. Let's suppose we imagine that we have a culture medium in which all 5 of them can be grown separately, or cultivated separately, right? So how it is achieved? How this is achieved? Actually, we make a liquid culture medium, and that culture medium is a special type of culture medium which help us to differentiate, all these 5 categories of bacterial growth visually. That we look at the culture growth. For example, we have a test tube in which there is special culture, and we throw all these 5 types of bacteria, or we have, okay, make it different thing. If we have 5 tubes, 5 different tubes, and in every tube one type of bacteria is inoculated, you will see in every tube different bacteria will make different growth pattern, which can be differentiated from each other visually. Such culture media are said to be differential culture media. What are differential culture media? Differential culture media are those culture media used in microbiology which help us to differentiate different categories of bacteria visually, right? Now, classically, the medium which is used here is called glycolate broth. The culture media which is used to differentiate among these group of bacteria is, glycolate, thioglycolate, thioglycolate broth fluid, or this is fluid medium, or simply we call it medium. So what is this medium? It is one of the, you need to remember that, because it is one of the very commonly used, very frequently used culture media in diagnostic bacteriology. I will explain that this kind of culture media, it is very important that you should have some clear concept. Why? Because very commonly, very frequently used in diagnostic bacteriology to inoculate the specimens and see that specimen had what kind of bacteria. Were there obligate anaerobes or aerotolerant bacteria, aerotolerant anaerobes or facultative anaerobes or microaerophiles and obligate aerobes, right? Now, because all of these 5 categories of bacteria have slightly different oxygen requirement and their behavior and relationship with oxygen is different, it means in this culture medium, within this culture medium, there should be areas with different oxygen tension. Let me tell you how we make it. Let's suppose this is a, this is a test tube, right? Suppose this is a test tube, and what are to produce a differential culture media which help us to differentiate these groups depending upon their behavior with the oxygen, right? We need a special type of culture media. And what is this? First of all, this should be a nutrient. It should have broth in it. First component, I will only tell you the most important component, the liquid which is present in test tube, right? For one component is broth. Now, what is the advantage of broth? Broth has many amino acids, many minerals, many vitamins, and it supports the bacterial growth. Broth acts as a nutrition supply for the bacteria. So, if this is having broth, then we will say this is nutritive. Yes, this is nutritive culture media. But with broth, we should have something else also. What is that? We should have, we should add to it, first we add the broth, right? We should add one more thing there, which should utilize oxygen and somehow produce anaerobic environment also into this tube, because we have to check, are there anaerobic bacteria or not? So to create anaerobic environment, we should add something to this culture media which will remove oxygen, which should make oxygen unavailable to the bacteria, right? We should make some area in the tube or culture media which is anaerobic environment. So we need to add a reducing agent, and reducing agent which utilizes, what is this? Utilizes the oxygen in this media, that is thioglycolate and cysteine, right? So I will say thioglycolate plus there is L-cysteine. Now, what we have done, that media which we are producing, culture media which we are now making, the important component are number one, broth for nutrition, for bacterial growth. Number two, to this broth liquid we add thioglycolate and or L-cysteine. These are two reducing agents which interact with the oxygen availability within this, right? And they neutralize the oxygen and make the molecular oxygen unavailable for the bacteria. That is why this is called thioglycolate broth fluid medium. Broth fluid medium. Thioglycolate broth fluid medium. Thioglycolate and cysteine act as a reducing agent, or if you cannot remember reducing agents, just remember thioglycolate and cysteine are acting as oxygen removal mechanism, right? And trying to produce what? Anaerobic environment without oxygen within the culture medium, right? This third thing is that we should add another thing, and that is oxygen indicator, because we should know what are the areas in the tube which are still having some oxygen, and what are the areas in the tube which do not have oxygen at all, and what are the areas which have slightly moderate amount of oxygen. Now, for that purpose, we add a special type of indicator, that is rasazurin. So rasazurin is indicator, oxygen indicator. When you add rasazurin into this tube, it will react with oxygen and turn into pink color. It means in this tube, wherever there is pink color, that indicates oxygen is present. And where there is no pink color, it means oxygen is not present, because broth and thioglycolate, especially broth, it makes a yellow color, right? Then we add thioglycolate and L-cysteine, right? Then we have also added to this culture medium rasazurin. The advantage of rasazurin is that it is oxygen indicator, right? And if there is oxygen present, this compound will convert into pink color, and visually we can see where oxygen is present and where oxygen is not present. And the last thing which is also important to mention, small amount of agar is also added. Why agar is added to this? Yes, small amount of agar is also added. Now there are four things added. Number one, broth is there for nutrition, thioglycolate and cysteine as reducing agents, rasazurin for oxygen indicator, an agar to reduce the oxygen penetration in the or movement in the tube, right? Now, these are the 4 important components. There are other components also, but these are the 4 you should know. Now, this medium which is made, what will happen to this? What will happen to this, that because above there is atmospheric oxygen, it means in upper part there is 21% oxygen, which is atmospheric oxygen. But as oxygen diffuses down to the media, it will be faced by the thioglycolate and L-cysteine, which will try to neutralize the oxygen, right? And as oxygen try to go down, it will get neutralized partially and then completely by thioglycolate and L-cysteine, right? And if above there is 21% oxygen, right, at the top, as we move down, oxygen concentration will decrease. For example, here is 20%, here maybe it is 10%, here oxygen is 5%, and here maybe 0% or 0.5%. The oxygen tension within, as you move from oxygen rich area, here it is atmospheric oxygen, 21%. As you move down in the tube, what you will see, that oxygen cannot diffuse up to down, because when oxygen from the top is going down, diffusing down, it will be neutralized or reduced by the thioglycolate and L-cysteine. And as you move down, oxygen tension become less and less, and eventually oxygen disappear from the culture media, right? Thioglycolate broth media. Now, when you add to this rhizozurin, what will happen? That upper part will be pink. Why it is pink? Because it is telling that there is, yes, more oxygen, moderate amount of oxygen, less oxygen, and no oxygen, because rhizozurin is turning into pink. So naturally oxygen will progressively decrease in tension and eventually disappear from the, what is this, test tube, as you go to the bottom of the test tube. So it means the top area will be aerobic. This area of the tube will be aerobic. Then we can say this area of the tube is having less oxygen. This is having oxygen nearly according to atmospheric pressure or tension, oxygen tension. Then oxygen tension decreases. It means we can say it becomes less than normal oxygen tension. So we can say this is micro aerobic environment, micro aerobic. And then it is the deepest part of the tube, that all very little oxygen diffuses down, and whatever oxygen is there, that is neutralized by thioglycolate and what is that, L-cysteine. So this will be anaerobic environment. What will be this? This will be an aerobic environment. So in this test tube, this culture media which we are using, right, we call it thioglycolate broth liquid media or fluid media, right? Basically this is present in a test tube. It is having broth as a nutrition, thioglycolate and sodium thioglycolate and L-cysteine as a reducing agent to eliminate oxygen. Rhizozurin is present as oxygen indicator. In the presence of oxygen, rhizozurin becomes pink. So upper part will be more pink. As you go down, pink color fades away, become less and less, and eventually disappears. And agar is there so that there is no excessive diffusion of oxygen from one area to the other area. In this way, this is a very special culture media. This is a very special culture media. What is the speciality? That within the media, we have developed differential oxygen tension, right? So naturally, bacteria which are in love with oxygen and can tolerate the oxygen, they will best grow in high oxygen area. And those bacteria, yes, those bacteria which can run metabolism anaerobically without oxygen, and they cannot defend themselves against reactive oxygen species, of course they will develop in bottom area. And after we see that as you move to deeper parts of the tube or in the culture, oxygen tension of keep on reducing, and there is a continuous gradient in loss of oxygen, that from aerobic, fully aerobic area, microenvironment which is fully aerobic, to it becomes partially aerobic or microaerobic, and then it becomes somewhat anoxic, and then it becomes completely anoxic. It means anoxic means no oxygen, it means become anaerobic, right? So now we are having culture media, if we are having suppose 5 tubes for these 5 categories, and in every tube we are putting one category of out of these 5, we will see that in such tube how bacteria make a growth pattern. Bacterial growth pattern will eventually depend, as I will explain in the next section. I will explain that bacterial growth pattern will be different in all 5 categories, and just by looking at the growth pattern we can tell that specific bacteria which is growing in the culture, is it obligate anaerobe or aerotolerant or facultative anaerobe or microaerophiles or obligate aerobes, right? After the break we are going to discuss. I will make 5 tubes and I will show the growth of all these different categories of bacteria, and I will tell that why every category has a different growth pattern, because every category deals oxygen concentration or tension in a different way. Every category will have in 2 ways difference. Some categories will love to use oxygen for metabolism, aerobic categories. Some categories which cannot use oxygen but they use anaerobic pathway, right? That is anaerobic category. And then we will also see how these different 5 groups of bacteria deal with the reactive oxygen species, and which groups are having full defenses, right? Antioxidant, enzymes. Which group of these bacteria are having dismutases, catalases and peroxidases, and which group they are defenseless, right? That they don't have such enzymes and they cannot grow near the top. Now you can understand, if a group of bacteria do not have defenses against reactive oxygen species, can they survive at the top? Of course not, because here is a lot of oxygen, right? But at the top only those bacteria will survive, right, which are having defenses against oxidative stress, right? Let's discuss after the break.